Hello, I'm Regan Jarvis. I'm the scientific founder and CEO of Anocca. I'd be surprised if any of you have actually heard of Anocca or myself. But we have been around for 5 years here in Sweden, since 2014. We're privately held biotech. Might surprise you to hear that we've raised just short of £500 million SEK to date, just closing around in June of this year. We're based in Södertälje at the ex-AstraZeneca facilities in Snarkviken, and we have a head count of 35 from 19 different nationalities, and the vast majority of those are scientific staff and manufacturing staff. So we have a human cell-based technology platform that's used for deciphering T-cell immunity, and that's a very broad-based technology platform, but we focus on TCR therapies. So we have a portfolio of deployable TCR assets for segmented TCR T therapies, and also technology that enables fully individualized approaches. We have our own GMP manufacturing facility down in Södertälje, and we will have our first segmented TCR Ts likely in the clinic by the end of 2020. So like I said, our focus is on TCR T therapeutics for oncology, and this is a stylized workflow for how that works. From a tumor, you have to be able to identify tumor antigen targets and then targeting TCRs with which to target those antigens. And of course, you need a product manufacture. This depicts an autologous manufacture where you recover blood from the patient. That's how we'll execute our initial trials. But the technology itself is agnostic. We can also take allogeneic approaches and have our own future developments in that area. TCR T therapies have been around for quite a while. Adaptimmune is the most notable mover here, but they've been slow to come to clinic. And they've not been widely deployed for a number of key reasons. One is poor target knowledge and high population segmentation, which I'll talk a bit more about. There's a paucity of good TCRs against even really well-characterized tumor targets. And that's a technological deficiency. And those deficiencies are lack of high-precision cell-based tools to read out functional aspects for segmented asset development, and also tools that can be deployed for rapid cycle analytics for fully individualized approaches. Now, trying not to get lost in the weeds too much with the T-cell biology, just introducing the challenges here, is that HLA diversity. So HLA is a human leukocyte antigen. These are the molecules which present antigens to T cells. And they're among the most diverse in humans, with thousands of functionally relevant variants. And that's for very important reasons for immunity at a population level. But the repertoire of HLA genes is essentially unique to each person, meaning that patients who have a given tumor antigen expressed in their tumor may present completely different epitopes to their T cells. That means that you get these highly segmented populations that you have to deal with. On the other side of the coin, there's the TCR complexity challenge. TCRs are generated de novo in the thymus every time you produce a new T cell, and they essentially get a random receptor. It's a recombination process just like antibody production in B cells. Healthy young adults will produce, give or take, 200 million new T cells a day, and the overall alpha-beta system, which is one type of TCR, has over 10 to the 18 possible combinations for TCR diversity. Coupled with the unique HLA repertoire within each individual, this means that the effective and safe repertoire of TCRs is essentially unique in each person. So to deploy any type of TCR T, you need to generate and characterize TCRs that selectively recognize the HLA antigen targets and some other factors that I'll mention further down. So the point is that neither antigen targets nor TCRs are universal. And this is the major analytical challenge for both asset development for segmented therapies, but also for the individualized approaches. And this lack of universality can be addressed in those two ways: segmented therapies or individualized therapies. And our technology enables both. So just to tell you what I mean about segmented TCR T therapies, you have a patient population. You can pass them through what we call immunoanalysis engines, platforms, and you get out just two things: HLA-restricted antigen targets and TCRs targeting those. And that builds up a knowledge base. Basically, you can define the addressable segment of the population, patient population, and provide targeting reagents, the TCRs, with which to target them. And there's a few key areas there. There are shared tumor antigen targets, like the ones you'll definitely have heard of, NYSO1, MART, things like this. Shared driver neoantigens, which are things like KRAS, BRAF, recurrent mutations that drive tumorigenesis, and viral antigens, which have a range of applications, both in transformed tumors, but also in infectious disease and some hematological conditions. So from this, you have deployable native, and in our case, engineered TCR assets for patient population segments. And this is certainly one approach we're taking, as I'll show further down. Importantly, our platform includes high-precision tools for TCR candidate selection, but also cross-selection for allo and cross-reactivity. For those who are a bit versed in T-cell biology, that's the major concern with the safety profile of these types of therapeutics. And we have a unique platform to address that. So on the individualized side, it may be obvious, but there is an individual. You must take input, diagnostic information, sequencing information, input it to your platform or analytical process in a customized manner to generate individualized data from where you can make your individualized medicine. And we're targeting TCR-Ts, like I said, T-cell therapies. Our technology, if you cut it in half, you can also address personalized tumor vaccines as well. And this will give a bit of a hint as to what we're talking about. I already mentioned we're using a cell-based platform. We've created a high-precision and high-throughput cell-based analytical tools, essentially representing an industrialized cell biology and molecular genetics platform. And what that means is that our core technology is based on libraries of highly engineered human cells. So we call it a plug-and-play engineered human cells to reduce the analytical complexity that I highlighted earlier. And how this works is that you have T cells and you have antigen-presenting cells, as in T cells, which are the main focus of T-cell biology, and those antigen-presenting cells can represent essentially any cell in your body apart from red blood cells and platelets. Now, there's all this noise, there's billions or trillions of receptors, TCR receptors, and a unique HLA profile for each individual. So when we create our platform, we use genome engineering to first remove those receptors. We've got now blank receiver T cells. These are immortalized cell lines of different flavors for different reasons. We also do functional engineering on both those sides to address different aspects of the biology, but importantly, we also insert standardized genomic cassettes. That means that when we operate the platform, we can insert our analytes rapidly at a pairwise manner, one at a time, for analysis, or in a library-based approach and cell-based arrays for different applications. And this creates your plug-and-play operation of those analytes, the TCRs, which is the main thing people think about, but also the antigens and HLAs. So you can make this highly standardized, reproducible analytical platform, which is very quick to cycle in an individualized context, or very high content, high throughput in a discovery development context. So creating therapies with this platform, you need an HLA profile of your patient or patient population, of course. You need sequencing data from a tumor, for example, or other diagnostic information like immunohistochemistry. You'll pass this through our cell-based tools of various flavors, and you'll get out, again, those two things. You get HLA-restricted antigen targets, you'll get targeting TCRs. Those are the two things you need to deploy these therapies. Now, looking at how we operate our pipelines, just conceptually, we have a standalone antigen discovery pipelines, and we address these in viral antigens, shared and driver tumor antigens, and individualized neoantigens. Those are the mutations that arise in tumors within individuals, which are essentially unique. These are all substantively linked forward into our TCR discovery pipelines, which include high-precision TCR identification, characterization, but also TCR engineering in a functional context, which is a unique technology that uses cell-based arrays. We're moving towards our shared tumor antigens into man by 2020, and I'll address some of those aspects further down. Just to give you a taste of our TCR Therapy pipelines, we have a range of TCR asset families against characterized shared tumor antigens. Some on there you'll recognize, of course, with precedent from others. We have shared driver mutation antigens, which we're very excited about. Some very strong assets in especially KRAS. In fact, as far as we know, these are the first human receptors characterized against this driver antigen target. And you might have heard a lot of hype around the KRAS 12C mutant-directed drugs recently. These would address actually a larger segment of the patient population of TCR-T therapies. We're also targeting viral antigens from different viruses on a range of restrictions, and this has applications in both transformed tumor treatment, but also some hematological conditions and viral reactivation in transplant situations. And finally, we're approaching the end of a proof of concept where we can spin this platform up in a completely individualized context, identify neoantigens from individuals' tumors, and produce TCRs that are effective against those neoantigens. I will highlight this top one, a top family of assets. These are against NY-ESO-1 and a not often talked about isoform called LAG-1, very similar. And these are shared tumor antigens, meaning they're overrepresented in tumor cells. Now, Adaptimmune have advanced quite far, and HLA-A201, which is the main Caucasian allele asset, through clinical trials. And our unique aspect here is that our platform is strong enough we can produce TCR assets against a range of restrictions. So we'll cover, rather than, say, 50% of Caucasians, we'll be in excess of 60% of the worldwide population. So at a very high level, this is our strategic outlook, is that today the whole field is here. We're treating few cancers with few people. We just don't have enough knowledge or tools to move forward. With our antigen discovery characterization platforms, we'll increase the antigen knowledge depth, essentially, so we can treat more cancers with more targets. It's just more antigen target knowledge. With our TCR discovery and engineering platforms, we'll increase that TCR depth and treat more people with more TCRs. And combining those two aspects, you'll get to a maximum practical segmentation of the population. So you'll become a point where it's going to be break even, how much you'll invest into asset development from what you can expect to sell on the market. And that doesn't stop us moving forward, of course, because we have this individualized approach. All those same tools, albeit slightly reformatted, are deployable for the sort of fully individualized therapies, where we can target individual neoantigens within patients, generate TCRs, and deploy those as a therapy. Importantly, to do this, we have our own established GMP manufacturing platform down in Sotelia, repurposed AstraZeneca facility, where the GMP infrastructure has been previously approved by the NPA. We've asked for this to be withdrawn because we're breaking down walls next month and reformatting and expanding that facility to cope with a few new processes for a redesigned production and some future scale. And this will see us through any foreseeable clinical trials for the next number of years. The infrastructure itself is modular. That includes the quality management system, so we can actually deploy a range of manufacturing approaches. There's a subtlety in the fact that segmented and individualized approaches will look different, in the fact that the individualized approaches will incorporate multiple TCRs in a given therapeutic, because you don't want to hedge your bets on just one neoantigen. We'll provide up to 4 or 6 TCRs in a single product. We're also deploying our in-house viral vector production for TCR gene transfer, which is actually a bottleneck in the industry at the moment. Aside from the cell manufacturing itself, getting your hands on viral vectors for transduction is a limitation. So looking at our near-term clinical pathway, at the moment we're deploying our first shared antigen assets into clinical development into 2020. And we're well into the manufacturing process development and validation at this stage. And like I said, alongside redeveloping the infrastructure to cope with those processes in the GMP environment. So product validation and clinical trials startup is on track for the end of 2020 and readouts in 21, 22. Like I said, a unique aspect here is that we'll be deploying on a single shared antigen target multiple HLA restrictions. And in fact, we have multiple TCR assets on each restriction, which gives us some flexibility for accounting for alloreactivity for those who are versed in T-cell biology. So we're also deploying in parallel technology into a GCP-validated format for fully individualized TCR-T therapies. This platform development technically is well advanced. The aspect that is most challenging here is digitization of the data management. So we have an in-house team of just 2 at the moment, coding up a platform to cope with the required data integrity and processing of that data. And we'll be expected to be ready to initiate those types of trials somewhere in 2021 or early 2022. I would say that, before I move to the last couple of summary slides, we did close a round in June, a significant private placement. We are completely privately funded, and we'll be looking to refinance mid next year to drive us into clinic. we expect to be clinical trial ready into Q3 next year. The shape and scope of that funding round will be decided based on how many of our assets we can out-license and the collaboration deals that we'll strike in the meantime. So we are interested in talking to investors, but it will be for around June, July next year. Just to mention some of our major research collaborators, have a fairly broad research collaboration with Janssen ongoing, and some local academic collaborators, including Rolf Kiesling, Lars Klarskov for rheumatoid arthritis, and Adnan Ashur at the SciLife Lab for some biophysics studies. Management team, I won't dwell on this. It's a broad experience in academia and in industry. And I will point out Hugh Salter, our Chief Science Officer, who's here in the audience and can answer any questions you might have over the rest of the day and tomorrow. So that's it from me.